Assalamu alaikum students. Today our topic of discussion is cell mediated immunity. It is the continuation of immunity. So the learning outcomes include discuss types and functions of T lymphocytes, describe how T lymphocytes interact with foreign antigen, and elaborate the importance of compromised immunity in AIDS. So we will discuss them one by one. Special attributes of the T lymphocyte system, activated T cells, and cell mediated immunity. Release of activated T cells from lymphoid tissue and formation of memory cells. Antigen, there is a T lymphocytes or lymphocyte clone and release T cells. These pass into circulation lasting for months or years. T lymphocyte memory cells are formed when a clone of T lymphocytes is activated by antigen. Many newly formed lymphocytes are preserved in the lymphoid tissue to become additional T lymphocytes. And when there is exposure of the same antigen again, there is a release of activated T cells occur far more rapidly and much more powerfully, because these are the memory cells and they recognize the same antigen. Antigen presenting cells, major histocompatibility complex proteins and antigen receptors on the lymphocytes, on the T lymphocytes. T cell responses are extremely antigen specific, like the antibody responses of B cell. B lymphocytes response to intact antigens. T lymphocytes respond to antigens only when they are bound to specific molecules called major histocompatibility complex proteins on the surface of antigen presenting cells in the lymphoid tissue. You can see here there is the antigen presenting cell which possess a major histocompatibility complex shown in this blue color, while the T cell has the T cell receptors, and you can see here the foreign protein of the antigen. So these antigen presenting cells present the antigen to the T cells. The antigen presenting cells include macrophages, B lymphocytes, and dendritic cells. The dendritic cells are the most potent. The MHC proteins are encoded by a large group of genes. Types of MHC proteins: MHC1 protein present antigen to cytotoxic T cells, while MHC2 proteins present antigens to T helper cells. The antigens on antigen presenting cells, while the receptor molecules on T cells. There are as many as 1 lakh receptor sites on a single T cell. Types of T cells and their functions. First is the helper T cells. About 3/4 of the T cell population are helper T cells, form protein mediators called lymphokines. Lymphokines act on other cells of the immune system and bone marrow cells. Lymphokines secreted by the helper T cells: interleukin 2, interleukin 3, 4, 5, 6, granulocyte monocyte clonal stimulating factor, interferon gamma. These are the lymphokines secreted by helper T cells, because the name indicates that they help in killing the foreign antigen. They help the T lymphocytes, they help the cytotoxic T lymphocytes in killing the foreign antigen. That's why they are called helper. Preprocessor area is exposed to antigen. You can see the major histocompatibility complex, and there is antigen specific receptor, and this is the helper T cell. So this helper T cell is activated there is also activation of cytotoxic T cells, and there are also suppressor T cells. These suppressor T cells actually suppress the activity of helper T cells and cytotoxic T cells, so that to limit their function only to foreign particles or antigen, and there is when there is exposure of antigen, there is activation of B cells and plasma cell formation, and these plasma cells produce immunoglobulins or antibodies, which include IgM, IgG, IgA, IgE, and IgD. So these are the immunoglobulins. Ig means immunoglobulin. So these suppressor T cells limit the function of helper T cells and cytotoxic T cells, and limit their role, so that they cannot act against our normal body cells. They only act against the foreign antigen. Cytotoxic T cells are killer cells. The receptor proteins on the surface of the cytotoxic cells cause them to bind tightly to those organisms or cells that contain the appropriate binding specific antigen. After binding, the cytotoxic T cell secrete perforins that punch round holes in the attacked cell. Then fluid flows rapidly into the cell from the interstitial spaces. Cell becomes swollen and rupture or dissolves. The cytotoxic cells also play an important role in destroying cancer cells, heart transplant cells, or other types of cells that are foreign to the person's own body. You can see here the cytotoxic T cells, killer cells. And there is the antigen is there. So there is the interaction of cytotoxic T cells with the antigen. Suppressor T cells are the regulatory T cells suppressing the function of both cytotoxic and helper T cells, preventing the cytotoxic cells from causing excessive immune reactions that might be damaging to the body's own tissues. So there is they have the role in immune tolerance. So acquired immunodeficiency syndrome, in the absence of lymphokines from the helper T cells, the remainder of the immune system is almost paralyzed. Helper T cells that are inactivated or destroyed by the acquired immunodeficiency syndrome virus, which leaves the body almost totally unprotected against infectious disease, therefore leading to the now well-known debilitating and lethal effects of AIDS. So there is the inactivity of helper T cells, or destruction of helper T cells by AIDS virus. So cytotoxic T cells, in the absence of helper T cells, the clones for producing cytotoxic T cells and suppressor T cells are activated. The lymphokines interleukin 2 causing growth and proliferation of both cytotoxic and suppressor T cells. The direct action of antigen to cause B cell growth, proliferation, formation of plasma cells, and secretion of antibodies are also slight without the help of helper T cells. Almost interleukins participate, but interleukins 4, 5, and 6, B cell growth factors, activation of the macrophage system, lymphokines are also there. So after a little bit discussion on AIDS, there is the immunization by injection of antigen. So dead organisms, chemical antigens. There is the example of typhoid fever, whooping cough, diphtheria, while The other way of immunization is by toxins, tetanus or botulinum, and there is live organisms attenuated. The example is smallpox, yellow fever, poliomyelitis, measles. So these are the different ways of inducing immunization by injection of antigens. So passive immunity by infusing antibodies, activated T cells, or both obtained from the blood of someone else or from some other animal that has been actively immunized against the antigen. So there is no challenge to the body. That's why it is passive immunity. Antibodies last in body of the recipient for 2 to 3 weeks, and during that time the person is protected against the invading disease. So this is all about the cell mediated immunity, the role of helper T cells, cytotoxic T cells, suppressor T cells.